Okay, part three. So this is part three of chapter 6, cells. We'll focus on the nucleus and ribosomes. So it's kind of important for you to understand the flow of information inside of the cell. So, and once again, when you understand these basics, you can take this information and then make assumptions and make judgments based on other larger things that are happening within the living systems. So, start with the nucleus. Nucleus is obviously found in eukaryotic cells only. It controls or it stores genetic information. 99.999% of the genes found in all cells are found in the nucleus. There are, however, some genes that are found. They're extranuclear genes that are found in things like mitochondria or the chloroplast. We've also discovered that in recent years that we've found exogenes that are found that just float around inside the cytoplasm of the cells, which is really pretty interesting. And it helps us kind of explain some really kind of strange diseases. So anyway, it's made of made of membrane. It has small pieces that are built into it called proteins. So here is our membrane here. A couple things I want you to know is if you notice, it said that it's a double membrane. So this membrane, the membrane is folded on itself. So typical membrane, which we'll get into in chapter 7 a little bit, is made of phospholipids. So there are two layers of phospholipids. This one piece right here is one two-layer phospholipid, and then it loops around and there's another two-layer phospholipid. Sometimes they call it the nuclear envelope. It's basically the idea is it's a hollow structure that is connected to something else. And in this case, if I don't know if I can zoom in here. There we go. If I zoom in here a little bit, you're going to see that here is our nucleus. Here's the nuclear membrane. All right, there's the space in between. This space empties out into the endoplasmic reticulum. So think of the endoplasmic reticulum as an extension of the nucleus. This is why you don't find ER in prokaryotic cells, because they don't have a nucleus. Let's talk a little bit more about these guys. So as I zoom in here, and I look at, there we go. I zoom in and I get a little closer here to the actual structure of the nucleus. There are these pores or holes. Right? The holes in the nucleus are to allow proteins to enter and leave, and more importantly, Mr. T RNA, mRNA to leave the cell or the nucleus. DNA does not leave the nucleus. DNA kind of stays in this. DNA stays in this area here. There's really two places where you'd find DNA. In this area, the nucleolus, which is where ribosomes or RNA is being made, and then it's in a kind of a stringy form called chromatin. So here you have a couple electron micrographs. This would be a scanning electron microscope, I believe. It's looking at the surface of the nucleus, and it's kind of, it almost looks like a crater on the moon. It's really kind of neat. And then there are these tiny little sections here. These are actually transmission electron microscope pictures of the actual pores. So what's happening is the pores are built of these proteins. Once again, you can see these little triangle-shaped things. These proteins then span that double membrane and create this hole. Things that are small enough can enter and leave through those holes. Once again, and those things are going to be RNA, Mr. T RNA. Okay. Let's talk about DNA. So DNA has there's a number of different terms you're going to find when you're talking about DNA. You're going to hear the terms chromatin, chromatids, TIDS, chromosomes, sister chromatids, homologous chromosomes. These will all be terms that you'll hear, and you have to be able to differentiate between those terms. Here's kind of a couple ways I like to think of this. So chromatin is double-stranded DNA, okay, that looks like spaghetti. So imagine like you have a bowl of spaghetti, and the chromatin would be the individual fibers or pieces of spaghetti. Chromosomes would be, if you wanted to eat that spaghetti, you would take your fork, take your spoon if you're a good Italian, go into the bowl, twist, and pull up kind of a condensed piece of, or strands of the spaghetti. That would be a chromosome. Chromosomes only exist during mitosis or meiosis, which only happens about 10% of the cell's life. So 90% of the time, chromosomes don't exist. They're in the form of chromatin or unwound spaghetti-like fibers. Okay, chromosomes, you've seen those looking like X's. We'll talk more about those as time goes on. But in typical cells, typical human cells, there are 46 chromosomes, 23 that came from mom, 23 that came from dad. You have numbered chromosomes. They go from numbers 1 to 22, and then sex chromosomes, X and Y. We number them 1 to 22, number 1 being the longest chromosome having the most number of genes on it, and then the pair X and Y at the end are your sex characteristics. If you have 2 X chromosomes, you're a girl. If you have an X and a Y, you're a dude. We can talk about issues with numbers there. There are diseases where you can have 3 X's, you can have 2 X's and a Y, you can have 1 X. Never is there a condition that I know of that you can have 1 Y, but once again, we'll get into that more. Nucleus, once again, there's a dark center in the nucleus called the nucleolus, or small nucleus, and the idea behind that is that's where RNA is made. Typically, it's ribosomal RNA, but we also make tRNA, which is transfer RNA, and messenger RNA. Those 3 things are vital to making proteins. We'll get into in the next couple of chapters how proteins are made, and Mr. tRNA is the big deal. So ribosomes are made of RNA. They're R, or ribosomal RNA. Ribosomes look like these tiny little dots scattered in and around the cell. Some of them can be floating around freely. Some of them can be stuck or bound to the ER. When you look at a ribosome closer, it's going to have 2 parts. It's going to have a large and a small subunit. They have numbers associated with them. For example, sometimes this is called the 50S unit, and this is called the 30S unit. The S, or the 50, or the S in the 30 stands for the number of base pairs found in that particular ribosome. For example, a 50S unit would have 50 somewhere in its number count of nucleotides, where 30 would have 30. And once again, we'll get into that as details kind of pop up for us. So ribosomes, free and bound. So there are 2 types of ribosomes. There's 2 types of ER. There's smooth and there's rough. Smooth ER generally synthesizes new phospholipids, synthesizes new internal membranes, synthesizes new membranes for the cell. It also produces hormones, steroid hormones. So cells like, let's say, in your reproductive areas would have large amounts of smooth ER because they're producing large amounts of hormones. Back to ribosomes. The ribosomes can be bound. If the ribosomes are stuck to the ER, and they're not stuck permanently, just temporarily, those bound ribosomes are stuck to the rough endoplasmic reticulum. Free ribosomes are free to float around in the cytoplasm or cytosol anywhere they want to go. Free ribosomes tend to make things that are going to be used inside the cell. So maybe it's a protein that's going to be used somewhere in the cytoplasm. That would be the responsibility of free ribosomes. And the ribosomes are totally dictated by the messages they receive. So let's say you punch a wall and your hand starts to swell and you damage the bone tissue and some of the bone cells. Those cells will be stimulated to unravel a piece of your DNA that deals with making new bone cells or new bone material. That message would be made in the nucleus. The nucleus would then leave the nucleus through a nuclear pore. It would find a ribosome floating around somewhere in the cytoplasm, and that ribosome would start to connect to and read the message and interpret that message into a protein. As this happens, as it begins, depending on the protein that's being made, it can either stay free, float around the cytoplasm, or it can attach itself to the ER. Bound ribosomes tend to make proteins that are going to be used outside of the cell. So those things can be shifted out of the cell and transported out of the cell through the endomembrane system. Another thing that can also happen too is bound ribosomes can make things that are going to be vacuoles. One of the most important vacuoles found in animal cells are lysosomes. Lysosomes are a specialized vacuole that have digestive enzymes inside. So imagine that you have this bound ribosome making a protein that's going to be an enzyme that digests the food you eat. You would not want that floating around freely inside of your cells, hence why they have bound ribosomes. All right, we're going to stop here. We'll get a little bit more into the endomembrane system. There's a ton of information, so make sure you're asking questions. You really need to watch it before class. Have a good day.